All right, good afternoon, everyone. My name is David Murdoch. I'm one of the CFAR members and helping Georgia out on the immunology core. And this afternoon's session is about introducing a few new faces to the field of HIV. We sort of began with that a little bit earlier with Dr. Kennedy's talk as well, and we're going to continue with some really great speakers. We're going to do a little shuffle here in the sequence. Dr. Shaw is, I believe, was coming from clinic and so forth. So, and Dr. Yellen got here a little early, so we're going to go ahead and start with Dr. Fox, who's going to be talking to us about the HIV reservoir in the kidney and the role of polyploidy. I must say, both Dr. Fox and Yellen are recipients of CNIHR awards, Creative Novel Ideas and Research Awards, who were very competitive award, and I understand about 11 of them were totally awarded, and North Carolina in total got 4, 2 here at Duke, 1 at NC State, and 1 at UNC, I believe. So we had quite a strong showing there. So I think that's great, and we'll get started.
So thanks very much to the organizers for this opportunity to speak in this session, I guess, about people who know very little about HIV to begin with. But I've really enjoyed working with Mary Klopman's lab on this project, and it's been a lot of fun. So I'm going to tell you today about a joint project on HIV reservoir establishment in the kidney, and we're specifically interested in the role of polyploidy, or cells with extra copies of the genome. So just to start with the very basic sort of way we think about building tissues or repairing tissues and progression through a normal cell cycle, there are normally the 2 key events that most cells will be doing will be replicating the genome during S phase and then splitting the genome during M phase. But it turns out that not all cells actually behave this way, and you can actually build tissue mass without any M phase whatsoever. You can just continually replicate the genome, and if you do this evenly, you'll result in a tetraploid cell. If you do it a second time, an octaploid cell. So these are polyploid cells. Polyploid cells turn out to be more common than most people realize. And so here are examples of polyploid cells in the human body. Many cardiomyocytes that are keeping your heart beating are polyploid. The cells that make your platelets, the megakaryocytes, are almost always polyploid. Cells in your liver that are responsible for making your bile and cholesterol, regenerating the liver, the hepatocytes become polyploid, and that frequency of polyploidy increases with aging. And the most extreme example of polyploidy in mammals are these giant trophoblast cells that nourish the early embryo. So it's actually a quite common and property that occurs in many important organs. Polyploidy is also present in disease, and we now know from sequencing of human tumors, of over 4,000 human tumors, that polyploidy is amazingly common. So 37% of tumors that have been sequenced have been estimated to undergo at least 1 genome duplication at some point. So polyploidy is thought to actually be a causative or pro-tumor factor in cancer. So my lab is very invested in this question. So we're fascinated by the fact that polyploidy has been appreciated since about the time that we've known that cells have chromosomes. Yet we still have very little understanding of some really basic questions, like why are cells or tissues polyploid? And then also related to this question is sort of an underlying need maybe to be polyploidy, is the fact that if you are polyploid, some of the rules for how a cell can operate are appreciated to have changed. And so we're also interested in what are sort of the costs and or benefits to being polyploid. And so just to give you a very brief background on my interests, most of my lab is working on fruit fly genetics, and we're interested in studying organ development and the role of polyploidy in that case. And so we study a Drosophila organ that is sort of addicted to polyploidy, if you will. So this is actually the fruit fly, the rectum of a fruit fly, and there are these 4 beautiful structures here called papillae that are critical salt-absorbing structures. And these structures are formed by division of cells with octaploid genome content, and we can show using genetic tricks in flies that if you make these cells now diploid, the organ does not form properly, and these animals have physiological problems. So it's a very convenient genetic system to try to understand places where polyploidy is really important. As an offshoot of this, we've also appreciated something that others have seen from systems as diverse as budding yeast and in cancer cells, which is that any time that a cell seems to duplicate its genome and then tries to segregate its genome, a polyploid genome during cell division, it doesn't do it very accurately. And so we found this to be the case in flies and in mosquito, and then around the same time that we discovered this was appreciated that in the mouse liver, so that was one of those tissues I mentioned to you earlier, when polyploid cells try to divide, they also make these mistakes. So sort of a recurring theme here is that when you have polyploidy, you're more likely to have genome instability. And one defense against this genome instability that's well appreciated is the DNA damage response. So I'm just going to give you a very brief overview of sort of the core regulation of that. When a cell encounters DNA damage, a kinase cascade mediated by the ATM and ATR kinases will then activate the CHEK1 and CHEK2 kinases, and then this usually converges on the p53 transcription factor to repair broken DNA, stop the cell from dividing with this damage, and also to undergo apoptosis if the damage is too severe. So this sort of is a balance against pro-genome instability factors. Now, where polyploidy counteracts this is that we've found that in diverse cases, including in cancer tissue, that the DNA damage response can be inactivated. For instance, in the placenta, CHEK1 is appreciated to be inactive. In the salivary glands of fruit flies, p53 has been inactive. And these findings have been found also in cases of viral infection, where polyploidy will occur. And so the bottom line here to appreciate, as I now sort of will switch gears and talk about HIV, is that when cells become polyploid in a diverse set of contexts, these cells then often become resistant to apoptosis, and they can also alter their genome. So it can sort of lead to sort of genome evolution within a cell population. And so I've been broadly interested in polyploidy, and as I briefly mentioned, we've also seen in the literature, it's seen that polyploidy is frequently found after a diverse set of viral infections. And so this sort of led to discussions, first with Mika Luftig, and then who then referred me to Mary Klotman's lab, who was actively working on a system where after HIV infection, polyploidy became present in the kidney. And so it's through work from the Klotman lab and others, it's appreciated that the kidney is an important reservoir in HIV. And so this came out of observation in the clinic that kidney disease can occur in high rates in aging HIV patients, and that even despite antiretroviral therapy, you can see the nucleic acid from the virus in the kidney of these patients, suggesting there's sort of a latent viral response here. And so HIVAN is one of these HIV-associated nephropathies or kidney diseases that they're associated with this viral infection. And importantly, in kind of getting back to this idea of evolution or genome instability, it's also been seen that in the kidney, you can have distinct evolution of these renal isolates, meaning that you can have viral sequences that's changing over time. And so where the connection to polyploidy comes in is that these patients with these HIV-infected kidneys can actually then become polyploid over time. And this has been seen from studies in the Klotman lab, both through human biopsies, where you can look by DNA fish. And if you notice in the control, we've got two different chromosomes here, and there's two dots, right, because you want to have one chromosome from mom, one from dad. But here in the HIVAN patient, you can see more than two spots for this particular chromosome, suggesting that in this case that there's tetraploidy that's occurred. And then it turns out that this HIV-associated nephropathy is actually very well modeled in the mouse. And so in mouse models of this, you can also get polyploidy. One manifestation of polyploidy are single cells with two nuclei, and that usually is the product of polyploidization by failure to complete cytokinesis. And you can see in this cross-section of a kidney tubule a binucleate polyploid cell. And so through multiple ways, it was demonstrated that after HIV infection and virus that can be transferred to the kidney can cause these kidney tubule epithelial cells to then polyploidize. So the Klopman lab has also identified the key gene in the HIV genome that is promoting polyploidy. It's the VPR gene, and VPR alone is sufficient to cause kidney cells to become polyploid. So these are human kidney tubule cells that were transfected with VPR alone. And what we're looking at here are FACS plots, and you can see here's a sort of control plot where you have the G1 and G2 peaks, but now in the VPR transfected cells, we have the third peak of higher ploidy, and so it's showing that VPR alone is sufficient to cause this polyploidization, and in the relevant cell type. And so to sort of summarize this, this is sort of where we started working together. We knew that polyploidy can lead to these sort of immortal cells that acquire genome alterations. The kidney is an important HIV reservoir, and VPR is promoting polyploidy in these tubules. So we're really interested in sort of investigating more, and particularly trying to understand how VPR is promoting polyploidy in the kidney. And, you know, sort of the most exciting interpretation of this is maybe polyploidy is actively promoting reservoir establishment or maintenance in this organ. And so the person who's really taking on this project whose data I'll show you next is a graduate student in Mary's lab, Emily Kam. And so one of the things that I've come to appreciate, so sort of coming from a cell cycle background where you usually have a very strict set of rules and controls, is that you know, when a host cell is seeing a viral protein, it's not really necessarily going to interact with just one of those controls. And in fact, that's what the literature has borne out. VPR is not doing one specific thing to a host cell cycle to cause polyploidy, and it's well established in the literature that it can do things like cause cell cycle arrest, cause cell death. It can do a variety of things to mitosis, and it can also promote DNA damage. It can promote DNA double-strand breaks. So I think it's important to have that appreciation when we're thinking about what we're doing when we express VPR in these kidney cells. And so Emily was really trying to understand how polyploidy is arising in this situation, and with the appreciation that it might be through multiple mechanisms. And so FACS plots can be really nice because they show you sort of the end point, but they don't show you the dynamics of the system. And so we took advantage of some really nice cell cycle visualization tools, such as the Fuji probes. And what these probes do, there's various variations of this, but in the one that we've been using, cells are red in G1, and then as they go to S, they become yellow, and then they turn green midway through S phase, and then you can see them lose the probe and become the cells will round up and undergo a division. And so here's a control movie on the left where you can just appreciate the cells change from green to red, and then you'll see the cells start to round up and undergo a division. And so here what Emily's doing is visualizing these mammalian kidney cells. And so then she takes these cells that are sort of relevant to the phenotype here and puts VPR in, and you'll see a very different situation. So these are sort of two stereotypical cells of what we usually see. The cell on the right is going to try to divide, and it's going to meet a very unhappy end, whereas the cell on the left will sort of round up and attempt to divide, but it will never complete the process. Right, so these are the two major phenotypes that we see over and over again when VPR is introduced into these kidney cells. And so another thing that's helped us understand what's going on is when Emily looks at the mitotic spindle, which is responsible for segregating the DNA, instead of seeing two nice spindle poles like you would see in a normal mitotic cell, the spindle, which is in green here, often has multiple poles. And so what that tells us is that the centrosomes, which are responsible for organizing the spindle, are probably multiply replicated, which happens frequently in polyploid cells. And if these multipolar structures go to completion, it's well appreciated that the DNA will just be ripped apart, create massive aneuploidy, and the cell will die. But what also happens is that cells that have these structures will decide it's probably not a good idea to divide, and they'll exit mitosis. And so we think that this is a reflection of what's happening in these cells here, and this cell was a cell that tried to divide with those multiple spindles. And so it's, of course, important to sort of quantitate this, and this is what Emily's done. And what she found is that 86% of her control cells divide, but only 18% of the VPR-expressing cells try to divide. And you can see this very strong correlation. The cells that die are the ones that really went through and tried to divide. The cells that live, for the most part, are the ones that had an incomplete mitosis. So now we've got a very good understanding of this protein that can do multiple things to the cell cycle, how it's promoting death in some cases, and then failure to complete division, and then continued cycling, which will lead to polyploidization. So given the connections between the well-known connections between viruses and DNA damage, and then DNA damage can sometimes promote polyploidy, it was important to think about the relationship between VPR and polyploidy in this context. And so what Emily's found are a number of important clues as to what's going on here. So she's found that viral integration is actually not critical for polyploidy. So if she packages the VPR-expressing vector in a virus that can't integrate into the genome, we see a similar amounts of polyploidy in both of these cases. So it doesn't seem like the integration step is really key here. Similarly, DNA damage alone does not seem in these cells to promote polyploidy. So these are cells that don't see VPR but do see two different DNA damaging agents, doxorubicin and cisplatin, neither of which are sufficient to cause this increase in ploidy. So it's not just that VPR, for instance, is integrating into the genome or causing DNA breaks that's sufficient to cause this ploidy increase. However, Emily's found that you can counteract VPR-induced polyploidy by blocking the ATM kinase, which, if you remember, is one of those upstream kinases in the DNA damage cascade. So when she adds a potent ATM inhibitor, now the frequency of polyploidy is decreased significantly. And so this leads us to our sort of current working model, which is that when these renal cells encounter the virus, there's sort of one of two paths. One is the decision to keep going, to try to divide, and these cells are probably loaded up with extra centrosomes. This is going to lead to multipolar divisions and death. But a number of these cells don't do that. They exit mitosis. They think better than to try to divide with multipolar spindles. They become polyploid. And then sort of where we're really interested in now is what does this mean for the kidney cells going forward? As I told you, polyploidy can promote sort of these immortal cells that are long-lived. It can also then, if these cells later try to divide at a later time, can lead to increased aneuploidy or genome alteration. And so for the virus, this could potentially be a mechanism that leads to those distinct sort of isolates that I mentioned earlier. Polyploidy is also appreciated to increase the amount of RNA or protein that a single cell makes, so it could also increase just the total virus that these cells are making. And in terms of what ATM is doing in this case to counteract that, well, ATM is well appreciated to induce cell cycle arrest, so it's possible that the ATM inhibitors are just preventing progression through that mitosis that would lead to polyploidy, and that would be a simple model. But it's also possible, and it's appreciated, that ATM itself can promote centrosome amplification, and so without that, you don't have that path that promotes ploidy. So we're interested in understanding more about what the role of ATM is and thinking a little bit about what ATM inhibitors might be able to do in the context of these HIV-infected kidney cells. So just to summarize, it's appreciated that the kidney is an important HIV reservoir, that polyploidy is a feature of the HIV-positive kidney tubular epithelium, that the HIV protein VPR alters the cell cycle of these cells in multiple ways, and that we know now that the way these cells are becoming polyploid is by failing mitosis. But we haven't found that DNA breakage is sufficient for ploidy, but we know that ATM is required. And we're really starting to think about now trying to get beyond sort of the description of how these cells become polyploidy and start to think about the really interesting questions of what might polyploidy be doing. Is it potentially enabling cell survival, viral latency, or further down the road, is it actually promoting resistance to therapy? And so I'll just acknowledge my partners in this, Mary and Emily. It's been really interesting to go from sort of my very basic science view polyploidy to thinking about this clinical application. So I'm really interested to hear what your thoughts are on what we're doing. Thanks. Right. So I think the distinction should be made between sort of developmental polyploidy and sort of aging-related polyploidy. So the answer is basically it can happen in both of those contexts. You know, the giant trophoblast cells become polyploid just as part of normal early development. So that's happening very early on and is a stereotypical program. Whereas it's not completely understood why in the liver, as you get older, the cells are more likely to become polyploid. I mean, I can speculate that, you know, sort of the liver is exposed to all sorts of toxins, and that may not favor undergoing a division if there's DNA damage. It's always safer to just replicate that genome within an existing cell, and so that may be one reason why that's happening. In the case of viral infection, you know, it's been shown in many contexts that some of these viral proteins can actually shut down the mitotic genes, but that doesn't mean the cell cycle has to arrest. You can still cycle without mitosis, and that's how you would get a polyploid cell in that context.
Do you think VPR is directly impacted, like physically associating with these multipolar spindles, or what do you think the molecular hook is to screw up what?
Yeah, so I mean, VPR has been implicated in, you know, different E3 ligase complexes that regulate the cell cycle, and some of those can impinge on centrosomes. So the ATM result is also interesting. You know, it's interesting to think about whether VPR might actually interact with ATM in some way, and without ATM, you don't have the ability to become polyploid, and it may be through centrosomes in that context.
Do the VPR positive cells have different responses to ionizing radiation?
I think that would be a really interesting and easy thing to look at, actually. Yeah, because that's something that's typically seen. Cancer cells that go polyploid become radioresistant. We see that in flies as well. It's very common. Yeah, that'd be a good thing to look at. Yeah. So the question was, is it only these kidney tubule cells that become polyploid? So there is a report in the literature of T cells becoming polyploid after HIV infection, but I'm not sure what. I think it's unlikely that every cell responds the same way. And I think, you know, some of these studies might need to be investigated in a little bit more detail, because sometimes if you don't, you know, look at things the right way with the right sort of fact settings, you might misinterpret some of the data. But what's, you know, as I alluded to earlier, it's only some organs that become polyploid, and I think, you know, the fact that this is accumulating in the kidney probably tells you something about the tissue type there. Yeah.